Kia ora and welcome to the second seminar organized by the New Zealand Marine Science Society. My name is Marika Babuda and I'm a student rep at the Society, and I'm going to be your host today. We initiated this webinar series as a replacement of our cancelled annual conference and to give our members the opportunity to showcase their science they have been doing over the last year. But we also organized it to open the doors to other ocean enthusiasts who simply want to know more about the research conducted in this country. We are a fantastic, diverse society, and we want to encourage everyone involved in marine science in New Zealand who is not a member yet to head over to our website and join it. We are a fantastic, diverse family. And this is especially attractive for students as we also offer a wide range of research and travel grants. Before we get started with our seminar, I would like to do some housekeeping first. So for all of you who are not familiar with Zoom yet, maybe there are some out there, on the bottom of your screen, you will see a little button with a label chat. So this will be activated for the next 10 minutes. And if you have any technical difficulties, our team people will try to help you sorting this out. This is a watch-only seminar, so your microphone and camera will be disabled for the whole time. But however, you can use the little button next to the chat button called Q&A, and you can ask questions to our speakers. So every speaker will be talking approximately 10 minutes, and after these 10 minutes, we have 3 minutes to discuss these questions. So I will read out the questions you're sending, and the speakers will answer them. If we have time in the end of all 4 presentations, we might also have a few more minutes to answer final questions. These webinars will be recorded, so they will be available on our New Zealand Marine Science YouTube channel. So please feel free to forward this information to friends and colleagues who are not able to attend today. Okay, let's get started. The topic of today's webinar is monitoring current novel approaches. And I'm delighted that we were able to invite 4 fantastic researchers today. And they will literally take us from the largest scale of monitoring possible to the very tiniest one, which is once again highlighting the beautiful diversity of the marine research going on here in New Zealand. Our first speaker is Dr. Lee Tate from Niwa Christchurch. Lee is a marine ecologist who is currently focusing on increasing spatial coverage of efficiency of ecological sampling methods to better understand the effects of local and global stresses. Without going into further detail about the amazing technologies Lee is using, I welcome Lee to the Zoom.
Kia ora koutou. I take it you've got my audio all good?
Yep.
And I'll share my screen now. And we've got a presentation live, correct? Cool. So today I'm talking about some emerging technologies that we're currently using to help us better monitor coastal environments. Informing robust management of our coasts and oceans requires knowledge of both stresses and the ecological, integrated ecological responses to those stresses. Doing this invariably requires measuring and monitoring ecological trends and environmental parameters very frequently and over large spatial scales. Unfortunately, we're quite far away from achieving this. So this is exemplified by some of the often talked about figures of the relative spatial scale that we've mapped our ocean, especially when we start comparing that to the surface of the moon and that of nearby Mars, which we've mapped in their entirety. Estimates put the percent that we've mapped the ocean at, various numbers. 5% seems to come up quite regularly, but I don't really think this does our scientific endeavors too much justice, and the story is essentially more complicated than that. If we look at a really coarse resolution, we actually have 100% of our ocean mapped, but that resolution is at 5 kilometers. That's how big the pixels are. While I'm sure most of you agree that 100-meter resolution, which is we've got 15% of the ocean mapped. I'm sure you'll agree that this resolution provides little relevant information for ecological purposes. I would argue that even a resolution of 1 meter would rarely allow us to identify species, let alone get a grasp of biodiversity metrics. So traditionally, our inability to consistently sample big areas of the seafloor has led to the need for subsampling, typically employed by quadrats and transects, drop cameras, and other spatially discrete techniques. And these techniques are really critical for high taxonomic resolution sampling. But the trouble in doing this is that it's very labor intensive, takes a long time to complete, and it greatly limits the area that we can sample efficiently. This also makes, this low spatial coverage really makes it difficult to detect meaningful change. And so this figure here covers quite a bit of ground, but the point that I'm really trying to convey is that achieving high statistical power, for example, greater than 80%, is really difficult, especially if we're trying to detect subtle effect sizes, so smaller effect sizes. And what you can see here is really that the message is that to get high power for very small effect sizes requires huge numbers of samples. And that really limits what we can detect and doesn't leave us much room for surprises. So the quest for meaningful ecological scale really comes down to expanding our observations, and much like those global 5 kilometre squared pixels of our seafloor, imaging satellites, Earth-oriented satellites, are imaging our Earth consistently through time and globally. While there's great promise and potential for these data sets to be used to help fill in some of the gaps in our knowledge, there's also great limitations to these data sets as well. For example, identifying small patches when your pixel resolution's up to 10 meters is challenging. It can be done to a degree. Identifying rare species is an enormous challenge. Identifying metrics for biodiversity, and as we all know, these imaging systems rarely penetrate more than a few meters into the water column, and that becomes reduced as we go into more turbid waters, which really dominate a lot of New Zealand's coastline. So emerging technologies, or at least improving technologies like aerial drones, can really help us fill in some of these gaps in spatial resolution. And this allows us to capture really high pixel resolution, and really comes hand in hand with the increasing use of automated detection algorithms, computer vision. So these high pixel resolutions allow computer models to identify, say, branching structures of a macroalgal species, or color profiles as well. These tools can cover, up to 1 kilometre squared reasonably if we're looking at an intertidal community. And I think this video exemplifies their ability to capture these environments under optimal conditions, which is really critical to getting the best quality data possible. So alongside object-based recognition, where we detect structure, we've also got spectral-based detection of species. The figure in the background shows a multispectral image of a rocky reef environment, and the figure shows the bands of 2 different camera systems. We've got a typical RGB camera and a multispectral camera. Now the multispectral camera captures discrete bands from the visible wavelengths, 400 to 700 nanometers, up into the near infrared, the non-visible wavelengths. And as you can see, they're discrete bands, they're not overlapping, whereas the RGB camera has just the 3 bands they're very broad and they overlap each other. And so these spectral signatures allow another mechanism of separating species, particularly vegetation, which give off really strong signals in the near infrared during photosynthesis. And so these three blue lines represent the bands that are visualized in this image. Unlike a regular image that we're familiar with, we can, which always has three bands, we have to view a multispectral image in three bands. And you can really see this near infrared band popping in the vegetation signals on this reef. So these multispectral images, here we've got one that was kindly provided by Marlborough District Council from Oliver Wade and captured by a third-party provider, Garden City Helicopters, who run a drone program. And there's four images here: a color, a RGB image, which is had a bit of a color stretch. We've got an NDVI image, which stands for the normalized difference vegetation indices. Basically gives us an idea of photosynthetic material. And when we input these layers of information into a automated classification routine, enhancing them with these valuable layers really helps the computer start to identify important parts of the spectrum that you've kind of identified to them. So they give a really good output in terms of identifying things like seagrass beds in this image here, and can easily separate that vegetation from both other vegetation like ulva or terrestrial vegetation, but also non-vegetated habitat like cobble. And so we're using these techniques to better understand the broad scale consequences of the 2016 Kokoda earthquake. And you can see these three images running from left to right show areas of increasing impact or increasing vertical uplift. And if we have a look at how that uplift has affected the relative cover of, in this case, bull kelp, general brown algae, which is predominantly carpophyllum, and encrusting coralline algae, we can see that most of these species dramatically decline with the level of uplift that occurred during the Kokoda earthquakes. And there is a north and south difference as well, related to a range of factors that will take too long to get into for this talk. But what the aerial imaging from drones has really allowed us to see is some of the areas that we can't necessarily get to during traditional ecological intertidal surveys. For example, bull kelp, the Villia species, were really heavily impacted by these earthquakes, and typically haven't occurred along the accessible fringe of the intertidal zone. But from our drone imagery, we're able to see that they still have survived on some further offshore rocky outcrops, which are subject to a lot more wave slop and have survived better through warm summers and hot days. So, of course, aerial imaging only gets us so far, and if we want to understand what's going on beneath the surface, then we need a different set of tools. And in this respect, we need to start sending things into the water. And this imagery shows some of the biodiversity captured beneath the Antarctic sea ice in a recent trip to Scott Base. You know, phenomenally stunning biomass of invertebrates and fish life as well. But this was all captured with a remote operated vehicle. And these next generation remote operated vehicles, or ROVs, have improved a lot of aspects relative to previous models. Particularly, they provide a highly stable imaging platform, which has really allowed us to provide flight routines, much like the aerial drones, which have very complex stabilization routines, which enable them to stay stable and fly grid-like patterns in an automated fashion. And this opens up several possibilities. So we've been using these stable platforms to fly grid-like patterns, much like we would with an aerial drone, and map relatively big areas of the seafloor. In this case, on the left here, we have an Antarctic benthic environment with a diver set out transect, but this has all been mapped with an ROV, and below that we have a digital elevation model, which is essentially a 3D recreation of the benthic environment. That's only possible by capturing that stable high overlap imagery. And there's a lot of potential outputs from this type of data. It potentially allows us to compare biovolumes through time and geolocate target organisms. And so we've, along another set of research goals, we've been looking at developing computer vision using AI algorithms to detect invasive species. In this case, the invasive Stylaclava occurring in Nelson Haven Marina. So you can see that there's a bit of current operating here, which the ROV is generally able to cope with to a degree, maintaining a stable, clear picture, very high resolution. And these automated detection algorithms, once they've been trained enough, can really help us to streamline some of those post-processing methods, which can be very costly for big video data series. So it's really a big goal of my research to try and increase the spatial scale of our observations so that we can better understand ecological processes. And as we start to increase the area that we're realistically able to sample, we can increase our chance of detecting more subtle changes in our environment, potentially reduce the chance of coming back from field research with insufficient sample size because you've discovered that the variability of your system is far greater than you expected. And larger spatial scales really do provide an insurance policy for being able to separate the effects of other events. And we see these all the time, and they're almost impossible to plan for. Not to mention earthquakes, heat waves, storm events. And we really need to, if we're going to better understand the interaction between local scale impacts and global scale climate change, then we really need to integrate over bigger spatial scales, over latitudinal gradients, and sample a bigger component of the ecosystems we're interested in. So all of this research has been really heavily supported by a range of collaborators and funding agencies, and I'd just like to take this time to thank everyone that's been involved and who I've worked with over the years and will enjoy continuing to work with. So thanks to all of you who have contributed to this. That's me, ready for questions.
Awesome. Thank you very much, Lee. And I already have 2 questions from the audience. If it will open. I saw they popped up. For some reason they're gone, but I remember one of them was if it's possible to detect mussel beds with the drone images.
That's a tricky one. I think if they're out of the water, there's a real possibility that if the drone's flying at the right altitude, that they could detect features in those mussels that are characteristic of them. I think that's highly possible. It all comes down to the altitude that you fly at, the lens you're flying with, and the type of organism that you're trying to identify. So mussels is definitely possible. It will require object-based recognition because I'm not certain there's a spectral signature like seaweeds that'll be useful. But there's definitely a clear shape to a mussel that I think an AI algorithm will be able to detect, providing that pixel resolution at the grounds is small enough.
Thank you. Another question is if you can determine percentage cover of macroalgae.
Yeah, totally. That's definitely what we've been working on. Part of it has been perfecting the spectral bands that we're using to detect those species, but once we've got enough validation data on the ground to relate the spectral signature that we're detecting from the air to a given species, we can be, to up to 90% certain that when we've classified it automatically, that's the species we've got, which is above what I've generally considered the threshold for high accuracy classification routines. So as soon as you've got that link between your spectral signature and your on-the-ground validation, then it's no trouble to put that pixel into a measure of coverage and then break up your image into chunks of interest and work out a percent cover or a total cover of that species for a given area.
Awesome, thank you. Okay, now we'll ask you to stop sharing your screen so I can invite our second speaker. Our second speaker is Dr. Robin Dunmore. Robin is a marine ecologist at the Cawthron Institute in Nelson, who is leading the monitoring of rocky reef habitats around New Zealand for the last 13 years. She has a strong focus in environmental impact assessments and monitoring involving aquacultural farms, port operations, and other coastal modifications that might influence the natural environment. Today, she will give us some insights into a long-term project she's involved with located at the Doubtful Sounds. She will share her experience in the development of this program and give us some insights and highlights of the data she has revealed so far. So the Zoom is yours, Robin.
Great. Kia ora, everyone. Thank you, Marika, for that. I'm just going to share my screen now. Just checking, making sure that that's okay. Right. Okay, so I'm going to be talking to you about the Doubtful Sound monitoring program that's operating down in Fiordland, and it's developed to investigate the effects of the freshwater discharge from the Manapuri hydropower station on the Doubtful Sound environment. So Meridian has, many of you will know, Meridian has a Manapuri power station on Lake Manapuri. It takes water from that lake, generates power as it goes through tail race tunnels and discharges that water into Doubtful Sound. And I just wanted to acknowledge straight up all of these people that are listed. There's a tremendous amount of work in this program that's been done in this program over the years, and I wanted to acknowledge all the people. I'll probably miss some out. I apologize for that, but in particular, the first few people that I've bolded there: Chris Cornelson, Eric Goodwin, Dana Clark, Paul Barter from Cawthron, Megan Carter, Graham Ellie, and Eric Stevens from Niwa, who are currently involved in the project. So the power station has been in use since 1969, and the monitoring has been occurring since 1997, first by Otago University, and from 2005, it was taken over from Cawthron and Niwa with Otago University support. We have physical monitoring that is going on there and biological monitoring. You can see from the map here, we've got the red triangles are oceanographic moorings there's 9 of them in total, 1 in Milford Sound and 8 of them in Doubtful. There's the yellow squares are meteorological stations where we acquire data on rainfall and wind. And the green circles are our biological monitoring sites. So the physical monitoring has oceanographic moorings, and they collect salinity and temperatures or temperature sensors at different depths to 19 meters. Our main ones are M4 here at mid channel and M1 in Milford as our reference fjord. These ones have full salinity and temperature sensors throughout the water column. The other moorings have surface salinity at 2 depths and temperature throughout the rest of it. For the biological monitoring, we monitor a suite of indicator variables and then try and determine exactly the effect of the tail race or other physical variables on these communities. So we have permanent quadrats set out at 11 sites, and I'll talk more about that later. We have permanent transects that NIWA monitor for black corals and sponges, their abundances, and also black coral sizes. We look at the vertical boundaries of species, so whether they're going shallow or deeper, and we do a lot in the intertidal with mapping and permanent quadrats. We look at echinoderm abundances because we know they can have a huge effect on the benthic communities, and we've done some soft sediment sampling. I'm not going to go through all of this, but I'm just going to pull out a few of the highlights and some of the changes that have occurred through time. So in terms of temperature, one of the values of these long-term data sets is you start to see some of the trends that are occurring. You can see here, this is temperature through the different, from the surface down to 19 meters, from 2005 through to 2018 at Milford and in Doubtful. And you can see here a few warmer years about every 5 years, and in particularly from 2016 to 2018, quite warm water occurring throughout the water column, not just on the surface. And that agrees with some of the literature that's come out that's looking at sea surface temperature rise from, for example, from Nick Shears and Melissa Bowman have published that, and from NIWA that have shown that there's been some marine heat waves. So it's the value of these long-term data sets. You really start to see some of these trends coming through, and we start to understand what the influence the tail race might have also on these temperatures. In terms of salinity, initially the program was looking at to determine the drivers of the low salinity layer that lays in the surface salinity, on the surface of the water, and how the tail race might affect the low salinity layer and deepening events. And this was done through looking at the long-term data set, and also with high flow trials where larger amounts of water was discharged, and they were looking at current speed, everything from current speeds to salinity throughout the water column. And what you can see here, this is salinity at M4 and salinity at M1. These bottom graphs show the salinity. So red is a higher salinity than the blue. At Doubtful Sound, we have the tail race discharge in Cumex. We have the wind and the rainfall, and the same in Milford Sound, obviously not the tail race discharge. And what you can see here is that there is a continuous low salinity layer maintained in Doubtful Sound, and that's the result of that tail race discharge. Whereas in Milford Sound, during rainfall periods that you can see up the top correlating with these deepening events and high winds, you have low salinity, and then at other times where there's no rain, it's higher salinity at the surface. And what's quite interesting, they're quite correlated through the different sounds as well. You can see these weather events coming through with deepening in both of the sounds. So we've got a good handle on some of the drivers of what's causing the low salinity, the continuous low salinity in the shallows and Doubtful are due to the tail race, but the deepening events are more affected by the amount of rainfall and the winds that are occurring. And with this knowledge, it's that we're moving towards more of a long-term monitoring program and using newer technologies. So what we've done with, I should have showed this one before. This is more recent data, but it shows the same sort of thing. So what we initially had with the salinity and temperature sensors in the sounds going back to 1997 was Odyssey sensors, which there were some issues with data quality and drift of these. These were changed by NIWA to RVR sensors, which were much more robust, and we've had some great quality data coming out since 2005. And one of the disadvantages with those is that they require 3 monthly downloads and servicing. So now we're going to be moving towards using seabird instruments, which actually contain an anti-foul, within the instrument, and they can be deployed for up to a year, which cuts down the download trips to annual. And you can see here, this is the design of the new floats with the sensors that are draped over the side down to 25 meters. These floats are also, they take the information from the seabird instruments, and Paul Barter has developed this for Meridian, and the data are telemetered through satellites. So the solar power and the wind power on these instruments allow that telemetry to occur. So we're not telemetering these at all of the data that's collected at a much higher frequency, but it allows us to check that all the instruments are operational, that the buoy is in place, it's got a GPS, so if it was ever to break loose, there would be an alarm. And it allows us to show to see all the power being generated by the solar versus the wind power to maintain that telemetry. So this is like a dashboard of what Paul's developed. He's got many of these moorings throughout New Zealand, and this is not just for looking at salinity and temperature. They're looking at different water quality, maybe turbidity or dissolved oxygen. And so when you look at here, you can dial into the dashboard and look at for vital signs, so how much power that it has. You can look at the meteorological data. These have a little wind turbine, wind sensor on them, and then you can look at the chart for the data. For example, so this is in Milford at M1. And it's at a depth of 7, and it's at the salinity that's occurring through there. So we can make sure that we can keep our eye on these instruments without going in there, and these are really remote areas to go in. So if it's quite an effort if something was to go wrong, and you had to go in. So having that check on the instruments through time is really good. And the bottom one just shows you the amount of wind power generated through time. So it just allows us to make sure that the power is being kept up so that the telemetry doesn't fail. And Paul's getting about 80 to 90% of the telemetry being successful. Sometimes on the satellites, these are really steep walls in these areas, and sometimes the satellites are in the right location for the data to be telemetered. But 80, 90% is pretty good, and they're telemetry data about once an hour. Moving on to some of the biological monitoring, I'm just going to talk to you about some of the first of the permanent quadrants that we've got set out in different areas. There were initially random quadrants through time from 1997 to 2003. In 2006, we installed permanent quadrants at 11 different sites and different depths in the intertidal for 2, 4, 7, 10, and 16 meters down with 4 replicates. So if you do the math on that, that ends up, you have a lot of photographs to go through. Paul's developed a lot of some nifty little ways of dealing with large data sets so that they pull the photographs into large databases, and it makes it much easier to handle things when you're analyzing them. And also just even renaming the files. He's got a little software program that makes it a lot easier for us to do that. So there's some great things that have come out of that. So what you get here is, this is a site in near First Arm, so near further down the fjord where you get more biodiversity, and near the Deep Cove at Site 6, where you get more Eurohaline organisms, and as you go deeper, you might have some black coral, but far less diversity than further out of the Sound. And what we've found through time, so going back to the exact spot on the rock, it's really valuable information. You start to see how long-lived some of these organisms are and some of the habitat preferences of some of the mobile invertebrates. So you can see here, this is at site 11 at 10 and 16 meters. through time from 2006 through to 2019. You can see a sponge continuing through time, some sponges here, and some algae coming through. So as I said, this generates a lot of images, which makes it very time consuming to analyze. We use a method of dots on rocks for this, where the images are brought into GIS, and 30 dots are overlaid on the image, and the person analyzes what's underneath each one. Phil Davies from the University of Canterbury is doing a PhD now and developing a neural network analysis. So it's taking these images, and instead of us manually going through them, he's teaching a network to identify these, using the data that we already have to train a network. So what he does is have, here's our 30 dots, and he takes 30 patches from around each of the dots, and then he creates a mask out of that, and then teaches that network to identify each organism. So there's not only the tail race or freshwater having an influence on these communities. We know there's a lot of other influencing factors. We saw last month with Nick Shear's talk on urchins changing the communities, and doubtful we'll see a lot of echinoderm effect as well. And one of them is from sea stars, which can be in very high abundances, as you can see from the photos here on your left. And so we monitor echinoderm abundances at our sites as well. You can see Dana here doing some quadrats through one of our sites. And so what we've found, this is a permanent quadrat at one of our sites. We were lucky enough to capture this predation episode where it started off with a lot of heavily dominated by mussels, but by the following year, nothing was left, and there's still no mussels at that site in those quadrats. There are some in the shallower ones, but it's really had a huge effect. And so knowing about the biology of some of these species is really important for understanding how they might affect some of the communities. Tracy Channon from the University of Otago did her master's on this, where she was tagging some of the sea stars. So she was using these little tags that were conductivity, temperature, and depth tags, and she was able to gather information about how far they're moving up and down in the water column, and what temperatures they were under, and also the salinity. So you can see on the top here. So each sea star is a different color: red, blue, or black. The one that lasts the longest, you can see the red in the red, sea star number 2. So at the end, there was a fresh with, you can see some fresh water occurring there, and a little bit of it dropping down. Also with the black sea star moving up into the shallows, you can see it was undergoing a lot cooler temperatures than the other ones. So this just helps us with understanding some of these processes that are going on and affecting these communities, and understanding that biology is really important. And I just wanted to highlight some of these little studies that go a long way in trying and understanding some of these processes. That's me. I just wanted to leave you with this quote from Kent Grange, which I think summarizes the Fiordland environment very well. And I'm open to questions.
Awesome. This sounds like a very intensive, big project, and I'm pretty sure everybody has like a handful of work to do on this. We have one quick question. Is there any pH data from this monitoring program?
Not that I know of. Not from the data that we collect on this. No.
Okay. And another one. Is there any planktonic sampling being done as well?
Not through this program. I think there has been done in the past, and some sort of snapshots of this, but in the past to determine what's happening with the plankton, but not long term. It's mainly this program is mainly focused on the rock wall, so the physical environment, and then the rock wall benthic communities.
Okay, thank you very much. Please stop sharing the screen, and I will introduce our next speaker, which is Dr. Vanessa Arranz-Martinez, and she is a marine scientist from the Mesa University in Auckland, specialized in molecular ecology. Vanessa uses molecular tools such as eDNA to investigate colonization paths of invasive species, genetic connectivity of New Zealand's coastal species to uncover barrier for dispersal and mechanisms driving spatial patterns. Today, she will present how she used DNA metabarcoding for characterizing the biodiversity associated with the eco-engineering kelps. Kia ora, Vanessa.
Kia ora, everyone. Let's see if I can share the screen while I'm saying how grateful I am to participate in those exciting seminars. It's everyone listen? Great. And thank you for letting me introduce also a new tool, DNA metabarcoding, to check if it's good for monitoring too. So as we've seen in the previous talks, traditionally, monitoring has been done using morphological-based surveys, sometimes using quadrants to estimate biodiversity. However, you see this stunning photo with a high diversity and taxonomically broad ecosystem. Those kind of approaches are being a bit hard because it requires a lot of time and taxonomic expertise, making it difficult sometimes to also detect critic and rare species. And it's hard to compare sometimes on different spatial and temporary scales, right? So metabarcoding, DNA metabarcoding has come and revolutionized the way we can characterize biodiversity. There are many studies that support that DNA metabarcoding is a cost-effective, transparent, and reproducible tool for biomonitoring. And it's such an emerging field coming up that new scientific journals have been created for those purposes, such as environmental DNA or MBMG. So DNA metabarcoding are different types, and you may have heard before. So the eDNA, that it's environmental DNA, is mainly collecting samples from the environment, and all DNA traces in the environment will be captured. While this community DNA, cDNA, is targeting certain bulk samples, targeting a specific community, right? Is the one that we will use in what I will show later on. So apart from where we take the sample, the metabarcoding protocol is pretty much the same. And it consists basically in different steps, a DNA extraction step, which extracts all the genomic material from the sample, a DNA barcode, which selects a specific part of the genome of the organisms, what is called barcode, with a PCR amplification. This barcode can vary this and can be designed for the different purposes. And then there will be a high throughput sequencing when we receive all the sequences that we will use later on to estimate the diversity or community composition of the samples that we are interested. Our study system is those amazing kelp forests. Kelps are ecosystem engineers and are responsible to support high diverse, complex, and productive ecosystems. And importantly, they are also sensitive to environmental change. We are particularly interested in this kelp holdfast, which anchors the kelp to the substratum, and it provides the home for a high diversity of organisms. It's also kind of a real opportunity to detect these changes in the ecosystem and responses to environmental change. However, despite this appealing use as a barometer for change, they have a lot of diversity, predominance of sub-body taxa, that it makes difficult for the morphological-based surveys and challenging to study them. So that's why we want to use to evaluate if DNA metabarcoding can help us as a monitoring tool for those systems. As we know, and we are aware, the morphological-based surveys carry some certain biases, such the size of the quadrants, number of quadrants of the observer. DNA metabarcoding is pretty much the same, also has potential biases, and it's crucial to consider how the decisions that we have in each of these metabarcoding stems will influence the inference in the taxonomic richness of the community composition of the samples. So in trying to evaluate that, we went to Mathison Bay in North Auckland to collect a few samples, and we collected a few kelps. And once in the lab, and thanks for the help of a kelp army processing that we will have to be really quick, we pull out the community from the holdfast, and we sieve it, and we filter it through 2 different mesh size to divide the community into a fraction of smaller organisms and a larger fraction of larger organisms. And we were particularly interested if those 2 fractions of the community show differences in terms of taxonomic richness and the composition of the community. We found that the larger fraction always, or most of the samples, show lower taxonomic richness than the smaller fraction. And we also found that both communities were different across all the samples that we found. And what it's also important is that if we extract the DNA together of the small and large fraction, like a no fractioning treatment, the taxonomic richness was always lower, and the community composition was different than if we extract the DNA from the fractions separately and we combine them in subsequent steps of that metabarcoding protocol. So mainly we saw that size fractioning is successful in reducing the misidentification in specifically the smaller organisms. Then when we go to the laboratory, there are many protocols that can vary according to the interest of the study. And we wanted to know if our procedural replicates were consistent and capturing a good representation of the community by specifically comparing the replicates in the extractions and the replicates in PCR amplification. We saw that the taxonomic richness was pretty similar across extractions, and it was a slightly difference in the PCR replicates. Maybe due pretty much for the stochasticity of the PCR amplification procedure. We also found that the community composition between extraction was slightly different. However, it was not different between PCR replicates, which highlights the importance of having more than one DNA extraction replicates. It's to compare like having more than one quadrant to capture all the diversity in a study system, right? And then the bioinformatic part analysis, it will start when we receive the DNA sequences, and I'm talking about millions, I would say millions of sequences, and they go through a quality filterings to reduce the number of false positives, reduce the sequencing error, or somehow contaminations. And then we can estimate the diversity and community composition of the samples. And we can do so at different levels. At ASV level, what is called ASV level, we'll be taking any different sequences as different taxa, where each different exact sequence will count for the diversity. But then we can also analyze the diversity at OTU level, which will take an arbitrary percentage similarity threshold to group similar ASVs. And that percentage will vary depending on the marker or barcode that we use to account for the intraspecific variation. Specifically for the barcode that we choose, that it was a CO1, 3% of intraspecific variation is expected. So we collapse the sample at 97% similarity. And finally, we can also infer the taxonomically, the diversity. We can also compare our sequences with reference sequence databases to find the taxonomic identity of the raw sequences. However, in that case, the diversity is normally underestimated. And that's because normally if we search our sequences against a reference sequence database, approximately like 50% of the sequences have not much found. Besides our efforts, we try to generate a reference database for marine eukaryotes for our study system, combining two of the largest repositories, that is GenBank and Vault. But besides our efforts, we found that the taxonomic inference are limited by the availability of reference sequences of the organisms. So there is an important need to increase the DNA barcodes and taxonomic curated databases. So maybe some of you are interested in comparing the metabarcoding results with conventional morphological based surveys. And to do so, we compare our OTUs assigned to a study that it was done 15 years ago. And to be fair, we used similar amount of Kelp holdfast diversity study and at a similar place. And we found that with metabarcoding, we can retrieve twice the amount of taxa than with morphology. However, as I mentioned before, the taxonomic accuracy is higher in morphology, especially at lower taxonomic levels. So it was more taxa assigned to a species level than with metabarcoding, and that's pretty much for the limitation of the reference sequence databases. If we look at the groups and proportion of taxa found with both approaches, some groups were equally represented with both approaches, like arthropods. Some of the groups weren't considered in that study 15 years ago based on morphology, and some were more successfully detected by one approach or the other. Like the soft body taxa are clearly better detected by metabarcoding, but the hard bodies like mollusk and bryozoa, it seems that it was all better with morphology, which means that somehow both approaches are complementary and it can serve to monitor these communities. So to wrap kind of all that genetic information that I give you, a few ideas to take home is that the DNA metabarcoding is feasible and promising tool to monitor this Kelp holdfast associated diversity. And if we wanted to do so, we have to consider the size fractioning and DNA extraction replicates to detect the wide variety of taxa. And as we saw, DNA metabarcoding retrieve higher number of taxa, but somehow is limited by the reference DNA barcodes. So I would like to finish just with a bigger picture of the project, because the main focus, it wasn't just to compare with morphological based surveys, but it's a project, a bigger project led by Dave Aguirre and Libby Liggins that we'll talk later. And I started being part of it and starting to participate in a bit of pieces of the puzzle, such as the DNA metabarcoding, to understand the diversity of the holdfast community. But it involved a bigger collaboration and many PhD students and master students working with the whole group. One part is to increase the DNA barcodes with collaborations with Otago Museum to complete the reference databases. And in the other side, there is also some students like Bo and Ho-Chan working to understand heritability of the holdfast traits that can be done by different means, like farming kelps to do breeding designs, or by molecular pedigree construction, to be able to understand the kelp phenotypes, the drivers of the kelp phenotypes, and ultimately link the phenotypes with the community created around there, and start understanding the eco-evolutionary dynamics in those temperate reefs. So as you can see, that's not a one-person work, it's not me alone, of course, and I have to deeply thank to all many internal help that we have at the uni, Massey, and many external collaborations that we received for this study. And that's me. Sorry for all that genetic fire, but if you have any question now, you can fire at me, and if not, you can contact me later on.
Thank you very much for this amazing talk. Even though we're running a bit behind time, I would like to answer, or like to ask at least one question from the audience. And it's like, did you also consider sequencing pooled PCR replicates versus single PCR replicates? For example, sequencing one sample that has 3 times PCRs pooled versus sequencing 3 times separate PCR replicates. Does pooling the replicates show the same overall diversity to the single PCR replicates?
Well, Yeah, that's an awesome question. And yeah, we have an experimental design and we consider that part. And it's interesting because when pulling, the PCR replicates in a single sequencing well, we retrieve a similar community composition, but lower taxonomic richness, especially at ASV level. If we examine the same at OTU level, the results are pretty much the same. So there are no significant difference between taxonomic richness or community composition, even if we pull all the PCRs, which make it a cheaper choice. So somehow we have a higher number of taxonomic richness at ASV level, but at OTU level, it can be the same and cheaper if we do so pulling the PCRs, and we can save money with that. It's a manuscript that we are putting together and hopefully we can send soon. So it's part of the study that we are doing with this experimental design.
Awesome, thank you very much. Please stop sharing the screen and I will go over to our last speaker. Our last speaker is Dr. Libby Liggins, who is also based at Massey University. She is interested in the processes that generate, maintain in the road biodiversity with a focus on the marine biodiversity of New Zealand and the surrounding regions of the Pacific. Her talk today is about the project Ira Moana, Genes of the Sea, and I'm handing over to Libby.
Thank you for the introduction, Marika. And thank you to NZMSS for hosting this webinar series. It's really great to be part of it. My slides up. Right, so today I'd like to talk to you about the Ira Moana or Genes of the Sea project, which among other things is looking to enable the monitoring of genetic diversity for New Zealand's marine populations. And when I say genetic diversity in this sense, it's different from what Vanessa just talked about, where genetic barcodes were used to understand what species are occurring within communities. Here I'm really interested in understanding what genetic diversity exists within populations and species. So why would we care about this level of genetic diversity and why would we monitor it? Well, just as we all know that species diversity contributes to ecosystem resilience and functioning, so too does genetic diversity or the composition of a population. So we know that genetic diversity contributes also to ecosystem function and resilience, and sometimes the magnitude of these benefits is on a similar scale to what we see at species level. Genetic diversity also seems to respond to pressures known to affect biodiversity generally, but over much shorter timescales. So for instance, a change in population connectivity, a change in population demography will be seen as a change in the genetic composition of populations through time, which may not be detected through population abundance surveys or species occurrences, for instance. We also know that genetic diversity in and of itself is important to monitor because it is really that raw material that populations and species need to adapt to their environmental settings and changes in those settings over the period of generations. And it's for these reasons that genetic diversity is considered alongside species and ecosystem diversity as being one of those levels that we need to safeguard, recognised within the International Convention on Biological Diversity. It is true, though, that we don't hear as much about genetic diversity as we do the other diversity levels, and there's probably lots of reasons for this, but I'll focus on a couple today that the Ira Moana project and some of my international colleagues and myself have been focusing on overcoming. So some of those challenges that we feel we're in a place where we're able to overcome them and see more monitoring of genetic diversity in future. The first one I'll kick off with is the fact that I think historically genetic biodiversity scientists, myself included, have been very bad at communicating with resource managers and those responsible for managing our biodiversity. We haven't broken down what are the important parts of genetic diversity to measure, consider and use as indicators. And as part of a large international consortia, the Group on Earth Observations Biodiversity Observing Network Genetic Composition Working Group, which is a combination of a lot of different conservation geneticist groups coming together across systems, we've been working towards this goal of breaking down what is it about genetic diversity that we really need to measure and monitor going forward. We have a manuscript in preparation that should be submitted shortly, and I'll just briefly cover it here in the hope that if you're interested, you can follow up shortly once it's published. But basically, we've come up with 4 different measures of genetic diversity we feel are important for monitoring going forward. These include an alpha diversity measure, broadly called genetic diversity, which includes richness, but also a measure of evenness, which is heterozygosity. It includes a beta diversity measure, broadly called genetic differentiation, a measure of inbreeding, which I think is quite intuitive to most people and we know has direct impacts on the fitness of an individual, but also a population. And the last one, which is a little bit more obscure, it's effective population size. So this is effectively trying to relay the number of breeders within a population. And this is important because it reflects something about the recent levels of inbreeding, as well as the evolutionary potential of a population in future. So it may look very simple, or maybe it doesn't look simple at all, but I guess it's important to take into account that this is a summation of a lot of work from a lot of really clever population geneticists over many decades. And really a group of us have put our heads together and pulled together where consensus is at to build this overview of what are the genetic diversity indicators we need going forward for managers. And it's been a really important debate and discussion to have, and particularly right now as we're in 2020, and right now our Convention for Biological Diversity is actually being redrafted. So what we hope is that lots of our messaging published in these manuscripts here, so several are already out, including our policy brief, and hopefully our final publication fully describing these indicators will be taken up in those discussions and potentially make it into some of the drafts for how the global framework will look to conserve genetic diversity going forward, both in terms of what indicators will be used and how targets will be set based on that data availability. So that brings me to my second point, which is really around monitoring the genetic diversity of New Zealand's marine biodiversity and where we really fall short at the moment. So it's true, as I said before, that genetic diversity is quite hard to monitor. We're now at a stage internationally that I think we have some really solid indicators that have a lot of support across nations, but we of course really need that data. So a couple of important things about the indicators we've developed. The first is that they are really applicable across all kinds of marine populations. So they work well for managed populations, harvested populations, captive populations, and also wild populations. The other point is that they can be calculated from any genetic biodiversity data we have for a population. So even in the case where we have a data set that was motivated by completely different objectives, not for monitoring the genetic diversity of this population, we can still use that data to now calculate these indicators and monitor the diversity of that population going forward. So we think it's a really great solution, but of course it really comes down to what data we have at hand. So the question of how accessible is genetic data can be answered somewhat in a convoluted way. So it's true that genetic biodiversity data is one of the most available biodiversity data forms that we have. And this is because there's been really great uptake by the community of open data sort of principles. There's always been a joint data archiving policy. Over the last 40 years or 4 decades, we've amassed a really large genetic data resource that it would be great to now repurpose toward calculating these genetic diversity indicators. And the amount of data we have is increasing all the time. So sequencing technologies are coming down in price. More and more people are looking to genetic biodiversity information. So we're increasing our amount of data all the time. This is putting incredible pressure on our data storage infrastructures. And it's become quite apparent to myself and colleagues overseas that we don't actually have adequate data storage infrastructure for the metadata that we really need. Okay, so we have genetic data, it's very accessible, but what we're lacking for the calculation of these genetic biodiversity indices is really the metadata that is needed to support the genetic data. So what I mean by that is that contextual information about the genetic data. We all collect it. It's something that we do in the course of our study. The most obvious one to mention is that of georeferences. You go out and take a biodiversity sample, you georeference, and you jot it down somewhere in a spreadsheet. And we collate this metadata throughout our study to the point of completion, where best practice is to deposit your genetic data, so often this is sequence data, into an open access repository. And this is something that's mandated by a lot of the top journals. We're very good at doing this, but we don't do anything with that precious contextual metadata, unfortunately. And the consequences of that missing metadata means that we have no ability to georeference that genetic data. We can't attach it to place, and we also can't attach it to community. So we can't feed back any genetic indices calculated from that data back to that community that may be custodians of that genetic or biodiversity resource or that place, and really those managers who have the most interest in that data. This is a real shame because around the world, people are trying to form a bit of a baseline understanding of what genetic biodiversity looks like. Where is it located? This takes, at the most basic level, a spatial analysis of these genetic indices. So without that spatial georeferencing, we can't do any of this. And although these graphs here, these maps look really impressive, I will tell you that in all cases, these are based on no more than 20% of all available genetic biodiversity information. So what that means is that around 80% of the genetic biodiversity data that is out there at the moment is actually not georeferenced in a way that it can be used to be spatialized or to calculate some of these genetic indicators that I've been talking about. How are we going in New Zealand? Well, part of the Iramoana project's mandate has been to look into this. And so we have found that there's around 530 genetic data sets for New Zealand's marine biodiversity, which is great. And about 265 of those studies are population genetic studies, meaning that there are one or more populations within that genetic data set that could be used to calculate these genetic biodiversity indicators that I was talking about earlier. So it would be great if we could calculate those and to have genetic diversity data sitting alongside mean species richness when we're looking at classifying our environments or maybe informing marine network designs. However, it's true of New Zealand as it's true across all of the world that although as a research community we've been really great at depositing our genetic data into these sequence data repositories, very well looked after, we've been doing absolutely nothing with our metadata. And this is not the fault of any individual researcher. It's really been a shift in what is best practice and an infrastructure need that hasn't been met in the past. And this is where the Iramoana project has really come in. So what we've tried to do is we've been working with several other groups to develop a data infrastructure and best practice guidelines to look after that metadata, which is essential for being able to reuse that genetic data going forward and to calculate and monitor genetic diversity going forward. So the Iramoana project exists within an infrastructure that we've built. So the infrastructure is called the Genomics Observatory Metadatabase. So this is purpose-built for the metadata that corresponds to genetic data, and it interoperates with the most commonly used sequence data repositories. It uses the latest standards or best standards that we have currently for biodiversity informatics and also for genomics data. And it interoperates with other biodiversity platforms like Barcode of Life Database and GBIF. Geome itself is queryable through a web user interface as well as programmatically through R. What's really original about the Iramoana project is that it's the first national implementation or uptake of use of this infrastructure. So it's been a good case study to look at what is required to mobilize use of this kind of infrastructure and what can be gained within a national setting if people start to use this infrastructure to look after the metadata and steward our genetic data into the future so that it can be used for monitoring going forward. So what I've been doing as part of the Iramoana project is been focusing a lot on education, so developing best practices and communicating about them, why metadata is important, why future use of your data will be really important to New Zealand as a nation, and also how to use the infrastructure. So the infrastructure includes a Iramoana team that sits within Geome, and within that you can easily download a template, a metadata template that you can use to store all your sample information or your genetic information. And alongside that, there's some recommendations that are bespoke to the Iramoana project that try to take into consideration, I guess, our biodiversity objectives nationally and our cultural and social context within which we work. Within the team at the moment, we have a lot of georeference data. I'd say that the gains in data have been quite modest, but there has been really good interaction with the community. So there's been about 90 people who've interacted with the project at different levels, whether they be genetic biodiversity researchers themselves or they be managers. And this has been across a lot of different institutions. So what I hope in the future is that people are using the metadata template and we'll see the gains from these activities in years to come. And for those who are new to the project and want to find out more, whether you be an end user or a biodiversity genetic researcher yourself, there's a website there that can help you interact with the tools and what the project's all about. So this part of the project is really about instilling that best practice, or having a conversation about what we want with our genetic biodiversity data in New Zealand, so that we're in a better position to monitor genetic diversity going forward as a nation. But what has also been a big part of the project is looking at those retrospective data sets, the existing data, and how we can potentially scoop that out, repurpose it, and make a bit of a baseline for our genetic biodiversity. So in doing that, I said before, we've highlighted a lot of studies that are potentially useful, and we've gone through the process of trying to georeference them, sometimes contacting the authors, and thank you to those who have got back to us, and to those who haven't heard from us yet, you might still. And what we've found or anticipate is that we expect about 93 data sets are going to be reproducible and reusable to calculate those genetic indices that I was talking about earlier. So what happens to these data sets is that once we recreate them, the metadata sits within Iramoana, and the data sets can be accessed and interoperable with that metadata. And they sit in a derived data repository, which we hope will be a dynamic resource that'll continue to be updated, and a resource for biodiversity managers in New Zealand. The repository sits along a bunch of other tools and data resources, the most important one being some code that helps you calculate those biodiversity, those genetic indicators for understanding the genetic health and viability of New Zealand's marine populations going forward. So in a bit of a summary, I guess, so monitoring the genetic diversity of New Zealand's biodiversity, it's always going to be a bit tricky to monitor, and it's less tangible than a lot of our other biodiversity measures. But we're lucky in that we're at a stage now where the international community, I think, has settled on some really good indicators. There's good momentum there, and we're lucky in New Zealand in that we have data, and we now have the infrastructure to make that data accessible. So I really encourage people to interact with me, whether you be a biodiversity researcher yourself, an end user, to let us know how the Iramoana project can potentially serve your interests, or how it can be extended to help serve those interests. It's been a huge project, lots of interoperating initiatives, I would say. There's lots of different layers to these sorts of conversations. So lots of people to thank, and thank you all for listening.
Awesome, thank you very much. I have one question from the audience, and it's, will you use the Iramoana framework to track new or invasive species coming into New Zealand waters?
Yeah, so I guess that question, it sounds like it's more of a species occurrence question, where I think there are other means to monitor species occurrences or new species occurrences, and one of my other projects is What's That Fish New Zealand, working with Irene Middleton, my PhD student, to try and set up an infrastructure that would help us monitor that through citizen science. I guess where it could be useful for invasive species is that, by monitoring the genetic composition of a population, maybe within a marina, an Ascidian, you have a population that's been surveyed 10 years ago, you go back and take genetic samples, you might recognize that there are new genotypes within that population, which would indicate that there's probably been a new reinvasion or some kind of admixture among populations, which is of course a bad thing, because genetic diversity is going to make that population more vigorous and potentially harder to deal with if it's a pest.
Okay, and one last one. What are your thoughts on microbial biodiversity in the light of this database?
Yeah, so my own entry into using this infrastructure and designing the infrastructure was very much from a population genetics point of view. Having said that, there are now other projects using the infrastructure, and they include microbial and metagenomic, metabarcode type data sets. So Vanessa's data set, for instance, is up there. And we have a publication coming out, so you'll see just at the bottom of your screen there, that describes a user case study. That was Lucy Stewart, actually, who applied it to a microbial data set. So here you will be able to see how she went about using the infrastructure and how it worked for her, and a personal account of the advantages and potentially disadvantages. So it's doable, and it's a dynamic infrastructure as well, I will say. I was talking to the developer this morning, and for instance, we've now implemented a new data field called name of individual, which means that researchers working with cetaceans, for instance, can now record the designated name to individual cetaceans. That often happens within stranding events I'm aware of in New Zealand.
Okay, fantastic. I think this will sum everything up very much. Thank you everyone for joining this webinar and for sticking around, even though we are a bit over time. Thanks to our speakers to taking their time and informing us about the research you have been up to for the last year. And I also want to invite everyone to join our next webinar, which will be next month on the 20th of August. And the topic will be tipping points, climate change, and unsustainable resource use. So if you have time, put it in your calendar and register for this webinar. And we also have some slots available for some of the topics. So please give us shoot us a text or a message if you're keen on joining this webinar series as a presenter. And I hope to see you soon next month for the next webinar. Have a lovely rest of your week. And thanks for joining again. Bye.